Hello and welcome to Proteins 101 with Elliot Simpson. I'm a junior biochemistry major here at the lovely Beloit College. And today I would like to give you a basics of proteins. What are their functions in the body? How are they structured on different levels? How this affects their folding? And how the differing charges within a protein are determined and affect the folding and function of the protein. So as we first start our studies of proteins, we want to understand what these proteins are doing throughout the body, what their function is, and what different types there are. So behind me, I have our 10 main types of proteins we're going to focus on today. We're going to start with our enzymes, arguably the most important protein in the body. Our body is a big chain of chemical reactions, so we want these chemical reactions to happen fast, as our body needs them to happen, and we want them to not take up as much energy. So these enzymes reduce the amount of energy needed for these reactions to happen and also speeds them up. Our structure proteins are used to give a framework for cells and muscles. Our transport proteins move molecules across the cell membrane or throughout the bloodstream, controlling what enters and exits the cells. Our storage proteins are used to hold on to nutrients, ions, amino acids, and then release them when the body needs them. Our hormones are used as signaling proteins. They act as chemical messengers that travel around the body to coordinate things like metabolism when your body's in stress. Our receptor proteins sit on the outside of proteins. They sit on the cell membrane, the surface, or on the inside of the cells, and they detect signals telling the cell how to respond to certain things that are happening throughout the body. Our contractile and motor proteins generate physical movement either in whole muscles or inside the cells, converting chemical energy into motion. Our defensive proteins are used to recognize and neutralize pathogens such as viruses, bacteria. These all keep the body safe. Our regulatory proteins control things like gene expression and cellular activity by turning genes on or off, adjusting the biochemical pathways. Our sensory proteins are used to detect environmental stimuli like light, cell chemicals, or temperature and convert them into biological signals for the cell. And that's your 10 types of proteins. Okay, now that we understand what the function of proteins is, why it's so important to us, and why it's important to our body, we're going to start to think about how this is built. We like to think of it as built on 4 different structure levels. To understand the second, third, and fourth, we have to understand the first, because that's going to build off the next. So we're going to start with one amino acid here. We're going to represent that as a circle. It's going to be bound to the next, and the next amino acid, and the one after that. The amount of amino acids in the chain is going to differ per protein. So these are bound by a special type of covalent bond we call a peptide bond. This is very important, and we're going to draw it all out with the different elements and chemical bonds. We're going to see how this peptide bond works, what the different kinds of amino acids are. There are 20 different amino acids that we care about in the human body, and they fall into 4 different categories. I'm now going to draw them all out on the board for you so that we can look at how that works. And there are all of our amino acids. Here are 20 amino acids within the human body. It seems like a lot. Don't be concerned. We have a lot of similarities, right? So we have 4 main categories. We have our polar, basic, acidic, and non-polar amino acids. This is just in reference to what they behave as in neutral pH in the body, whether they're basic, acidic, act as polar, or non-polar. So, as you may see, all of these have a similar structure, right? So they all have an H2, OH2, and a C double bonded to an O. If we circle this right here is asparagine. This past here is called our R group. That is what changes in all 20 of these amino acids. If we look at our polar groups, we have a lot of C double bond O's, OH, SH, OH, right? So these are going to give us polar interactions when interacting with other chemicals in the body. Now, like in our basic amino acids, what basic means is it just means that at a human pH, which is about 7.35 to 7.45, these two, lysine and arginine, are going to want to gain a hydrogen. That means this is going to turn into an NH3 plus, and they're going to have that positive charge in most cases in the body. This is kind of the reverse for our two acidic amino acids. These are glutamic acid and aspartic acid. These are going to want to lose the H on the OH here at the end of the chains. It's going to give them a negative charge at human pH. We then have our nonpolar amino acids. These are going to have nonpolar interactions with other chemicals in the body. So one of the reasons that nonpolar and polar is very important within the protein. If you think about our cell, it's wrapped with the cell membrane. On the outside is polar things like water, other things that dissolve in water because they're polar. That means that these amino acids are going to face on the outside of our protein to deal with those polar things, while our membrane, it is built of two layers. And on the middle, we have a non-polar region right here. That means our amino acids, if we have a protein in the membrane, are going to want to face on the inside and deal with those non-polar interactions. So now we're going to move on to thinking about our second level of our 4 levels of structure. Our secondary level of structure can fall into 2 things: our alpha helices and our beta pleated sheets. We're going to use some Greek symbols for alpha and beta. An important thing to remember in understanding what these are and how they work is that they're driven by hydrogen bonding. When we think about what our amino acids look like, right, we had an NH2, C double bonded O, and an OH, right? This is what every amino acid looks like at a basic structure, and then we have our R group over here. This H on the end is going to interact with this O over here, because we're thinking about how we have this long sheet of different amino acids. So when each of those O and H's interact, we get what we call hydrogen bonds. This is going to drive our alpha helices and beta sheets. An alpha helix looks like a coil. It's hard to draw two-dimensionally, but it's going to be a coil, kind of like this. Beta pleated sheets can either be parallel or anti-parallel sheets. They can be, you can think of them kind of like a block like this, kind of running just like that right next to each other. And so here we're going to have our hydrogen bonds between these neighboring amino acids right here. Because remember, these are all amino acids in here. On our helices, every 4th amino acid is going to have a hydrogen bond to the one after it, right? So we're going to have all these amino acids, they're going to hydrogen bond, giving it either this coil or these arrows. And then these are combined by what we call a disordered area. It's just a strand of amino acids that doesn't partake in secondary structure. So we might have that, then to this, and then it's going to come back. Maybe it ends up here, right? That is our secondary structure. So now that we understand primary and secondary structure of proteins, we're going to look at the 3rd level of structure. This is the 3-dimensionality of a protein. So here we're going to have our area of randomness, a helices, then area of randomness, maybe another helix, area of randomness, then maybe we have some beta pleated sheets over here. We'll go parallel. Now we're going to think about how these all interact with each other. So keep in mind, we have water all around out here, right? A whole bunch of water molecules. And the important thing here is we thought of secondary structure as driven by the hydrogen bonds. That's what forced them into these pleated sheets or alpha helixes. So we think of bindings and bond as enthalpy. That's what we call enthalpy. Here, we're going to think about how much randomness do we have? Entropy. We're going to think about the randomness of the water. So when it's all spread out like this, we're going to have water in here, and in here, and in here, and in here. But when we take three-dimensional structure and we bring it all together, so this coil is going to interact with this beta pleated sheet, and it's going to move in this three-dimensional kind of bundle, right? So then we're going to have our helix, and here with another helix. Obviously, we can't draw it on a two-dimensional board. So maybe this helix is here, we have another helix here, beta pleated sheet between them. It's all going to kind of ball into one. So when this happens, we have these waters getting kicked out from in here to out here. It's called the hydrophobic effect. So we can think of the third level of protein structure is driven by this entropy, this degree of randomness. Because when we get the water out from in here and out here, we're going to have more randomness, more different configurations you could have with these water molecules on the outside. There's more possibilities for how they surround the protein than if a couple of them were in it and we didn't have as many out here. Okay, now that we have started to look at the three-dimensionality of a protein and how it works, we're going to now look at the fourth degree of structure. That is, if you noticed how second degree, it used the building of primary, right? It had the amino acids, it made the coils and the sheets. Then our third degree built off secondary, right? It was how those secondary structures interacted with each other. We're going to do the same thing here. So here we have our one protein, we have one unit, right? This is our tertiary structure of protein. Then we have another one. So within this, right, it's all a long chain of amino acids. It has alpha helices, it has beta sheets that are interacting with each other to ball up. We have areas of randomness. So here in our quaternary structure, we're going to use two colors to indicate this. Here we have our black subunit, our blue subunit. So say our black one is going to interact with the blue one, right? So now they're going to stay separate. Within each one we have our amino acids, our secondary, and then our tertiary structure. These are going to interact with each other. We're going to make one big protein built of two what we call subunits. They are different tertiary structures that are going to then interact with each other to make our one big quaternary structure. And now you know how proteins are structured and built. So now that you understand the function of proteins and how they're built, I'd like to present one last idea. This is the idea of denaturation and native fold. So our native fold of proteins is going to be the configuration of this protein on all four of our structure levels that is the most common, what the protein wants to be. An idea of denaturation is, say we put a protein in a pH or a temperature that it doesn't like, right? It's too hot, too cold, too acidic, too basic. It's going to start to denature. It's going to start to fall apart. In extreme levels, that is the amino acids falling apart, getting cut up, those peptide bonds being cut up. We could also have our things that hold together our secondary, our tertiary, our quaternary structures. The protein starts to fall apart. This means that it's going to not want to be in the native fold anymore. It can't be. It can't be in what it wants to be. This can be thought of by looking at here we have, okay, maybe this is a fold. This is a possibility of it folding, right? And then it's here, and this, at the lowest energy, with the most stable, if we think about this as stableness, this is our native fold right here. This is what the protein will be at ideal conditions. And then maybe it has different possibilities. But this is giving our native fold. And that is all you need to know about proteins to be dangerous. Good luck in your science studies, and thank you for following along with me.